Good afternoon, everyone. It's my great honor to present our work. I'm going to talk about the organs and chips that we study the immune cell interactions and also report some of our recent progress in our lab. For the background, we all know that for the drug screening, we need a better model. Of course, a 3D model is more about mimetic, especially with the microphysiological conditions. And also for the precision medicine, in fact, it also requires a better model. Like we can see there for the FDA, it shows that for the patients, for the cancer treatment, its effective rate is only 25%. This is ineffective rate. So basically, the patients may also need an organs on chip as its avatar. Apparently, the gene sequencing can help a little bit, but not to all the patients. And also the PDX can help some of the patients, but with its low efficacy and high in expense, it's not very ideal. So that's the reason emergence of organs on chip. And we can see that for the US EPA, it announced in 2019 that it will eliminate all mammal testings and choose organs on chip as an alternative. And in China, it's quite similar. China says for the samples that they don't have the good animal models, they accept the organoid and microfluidic model as a validated model. Especially after last year, the FDA Modernization Act 2.0, then everyone is so agitated about organs on chip. I don't need to go over the history of organs on chip. Apparently, it's very important, and it is the top 10 technology of 2016 by the World Economic Forum. And organs on chip has been developed both in the United States and in European extensively. And also its application has included environmental evaluation, disease modeling, space medicine, and drug discovery. In our lab, because we're from Southeast University, so we're in China, but we do also participated in a lot of organs on chip research, including develop microfluidics, materials, and do the biosensing, and also do some developmental biology. In fact, when we're tracing back to the history, our state key laboratory of bioelectronics takes the lead in organs on chip research in China ever since 2011. And then we got some award. We got the innovation research team at the Ministry of Education of China. And then we gradually developed all different type of organs on chip, and we also got the national key R&D program in 2018. And later on, we developed the first space tissue in a chip in China, and also we developed initiate the first OOC society in China. And we also got the first Chinese standard skin chip. It's now in public. So while we're doing the research, actually, we also have quite a lot of connection with the world. Like we invited Dr. Dan Taylor, Dr. Cam Liang, and a lot of Dr. Mark Texter, and a lot of the famous researchers in organs on chip research, and visited China and also visited our plants. And they have given us very useful suggestions. And we also went to Switzerland and a lot of different countries to participate in the world in the activities worldwide. So for our studies, on one hand, we develop materials like the materials with holes, or the materials have alignments, have directions, and direct the cells, cell contact guidance, so form certain structures. We also make electrospin fibers and for better cell growth. At the same time, we also make like the dual network or multi-network hydrogels to make the ECM more stiffness adjustable, so it can be more similar to each tissue. And we can also use light-controlled re-editable surfaces to change the surface molecules, and also we as the last speaker said, we also do the laser, basically it's 2-photon printing. So now we can print materials that to reach a level around 150 nanometers. So it's a scale that's quite close to the cell sensing's lower edge. We can also adjust the stiffness or the structure of the material to make it to have different colors. So it's called photonic crystals. And using 3D printing, we can also make the surface more hydrophobic or hydrophilic. But with the photonic structures, we actually can put cells onto the surface. And when the cells exert force on the surface, it will change its color. I will show that later on, but just go over the technology at first. So since 2012, our group have published more than 100 SCI papers in the organs on chip research field and received 90 plus patents already. For industrialization, we developed like the tumor on a chip. So we make the kit and we make the chip, so the customers can use our kit and chip to directly make tumor spheroids and tumor organoids on the chip. And we also have the called a smart system. It's a high content imaging system and the AI analysis to minimize the user's time and to maximize the efficacy. We culture the cancer organoids, and they're quite similar to the original cancer, and all those are tested in the chip, and we find the can monitor the different drug efficacy. More interestingly, we can see that for the primary cancers, if they are non-invasive, during the medical testing, we find they are not metastatic, they don't form the invadopodia. But for the medium invasive or high invasive cancer patients, we can see they form a lot of protrusions, invadopodia, into the ECM. And also we can see the necrotic core that inside of the cancer spheroids and cancer organoids that we made. So on the left side, it's an invasive cancer. You can see a lot of cell migrate. And on the other side, it's non-invasive, but it has very clear borders. So we have the high content imaging, the pumping system, and the environmental control system. So this one we have supplied to different hospitals, and the doctors find it very useful. Also, we have the software to distinguish the organoids individually and to measure against time. So then we can consider each organoid as a separate testing sample. We also tested CAR T and a lot of different drugs in our system. They are comparable to animal tests, and some are even better. We also evaluated targeted drug. And so all those tests are with big pharmas in China. So for the tumor side, we can develop the tumor microenvironment and also include the flow mechanical property and also different cells together to construct a customized OOC system. Besides that, we also made some heart on the chip. So we do the screening for Henry, and they have developed the first novel cardiac drug, and it's already under IND to the CFDA. And after 6 rounds of screening, we decreased the compound IC50 to 1/4 of its initial stage. And you can see these are the cell sheets, these are the cell spheroid, and we also have cell bundles that can attach to the 2 cantilevers on our chip. We also made different type of disease models, and in fact, after different stimulation, you can see the functionality getting better for the spheroids. We also developed AI cardiac organoid video analysis system. It can distinguish a different organoid and measure at the same time, and also they do the calcium imaging at the same time. We also use some technology called light field microscopy, so that you can see those bumping of organoid from one place to the other. It's actually a 3D image, so it gives us a very good tool to analyze where the pace started. And we can also measure the cell forces. So basically for the organoid forces, we use a substrate, as I mentioned to you, the photonic crystals. We can measure the hard spheroids. So basically it's actually like a 500 millimeter spheroids, but after drug treatment, it force significantly decreased that we can very quantitative measure. The colors directly reflect the force. We'll also do some liver on chip and skin on chip, including epidermis, melanin skin, and also the full thickness skin. So based on that, we're also using the OECD standard to do different tests. The evaluation including its structure and also like the resistance value, its a tier value, and also the permeability of the skin. And we tested the OECD 439 samples and find our skins correspond correctly, exactly to the test samples. We also tested the permeability things, which I wouldn't go any further. We also make some melanin skin chip. So you can see this was without and this with, and we tested different chemical samples. Some are from the cosmetics companies. I found it's very useful to evaluate their products. We also made the full skins. So these are the full skins with Transwell. These are the full skins that we made with our chip. You can see it's thicker and a better structure, and this is human skin. So we compared them. The same for the skin. We have the chip and we have the fluid system, and we have the analysis system. So basically combine them together. A cosmetic company can do the test in their site. We also have the standards of the skin chip in public. So we will have about 12 months left, and then it will be finally valued by the Chinese Standard Committee. Hopefully, we can get the first national standard in China. We also use the machine system in the CDC, and now currently the Jiangsu CDC has using our devices, the high content imaging devices, to test more than 500,000 samples. So it shows the automated system, how it can help CDC and help people. So about the topics that interactions between the immune cells, I'm going to talk about when we are making the lung chip. So this is our title, a storming a teacup study on the spreading and inflammatory cascade reaction of COVID-19 and on the lung chip system. So we use the saying in English, say storming a teacup means no big deal. But if we consider storm as cytokine storm, we consider teacup as organs on chips, then it indicates that we probably can monitor the inflammation inside of the chip. So these are how we can make the chip. So the chip is pretty easy to make. It has two channels or one channel, two chambers. One is the alveoli and one is trachea. And we also have tier integrated into the chip. So it is quite standard for us how to make a lung chip. We have the lung epithelial cells and we have the lung endothelial cells, and we measure the tier value, we measure their cell-cell junction, we see their morphology. And besides that, we find if we include immune cells inside, like the macrophage. So those macrophages included, you can see it attached on the epithelial cell layer. And also we have circulating the leukocytes, including monocytes, and later on we include other immune cells. And those involved of these immune cells altogether have interesting chemical reactions. And we found if we put LPS only inside of that system, although there's an elevation in some inflammatory cytokines, it's not that significant. But if we have the LPS and the monocytes and the macrophage, and then significantly increase for the inflammatory cytokines. We have 3 measured. I didn't put it there. It's IL-1, MCP, and TNF-alpha. So basically, some of the inflammatory cytokines can increase more than 50 times or 100 times. So that significantly increase, showing the inflammatory amplification. So with that, we can also measure the cell permeability on the chip. We have measured the bronchial chamber and alveolar chamber. They all have significantly changed. And we also developed a perfusion system ourselves. So basically, it has the pumping region. Those are 4 chips on the upper side. We have also downstream 4 chips. So basically, and we have a perfusion system and a nebulizer. So basically, we can form the foam and from the upper layer to the lower stream. So in this setup, just like schematically showing here, we will be able to tell if a virus or a bacteria can infect, say, from upstream to downstream. If upstream is an animal, downstream is a human, then we can answer if it can be transferred from animal to human, or if it can transfer from human to human. We tested the colors like pigments, and we also tested the virus and the proteins, and we find it have a good mimic of the transfer of the particles between human. And in order to measure the differences, as I said previously, we can measure the activity, the permeability. But besides that, the images also tells a lot. So those are the inactivated macrophages. You can see they're round, they're circling around, they're moving around. But once the activated macrophages, they become flat, and they do the searching, and they do the penetration. They have the amoeboid-like migrations. So from those images, together with the endothelial images, epithelial images, and the images from other immune cells, for all together, we can have a very clear cut-through value for the distinguish of infection or not infection. So those are the training numbers. With the increasing training numbers, it becomes very quick to be accurate. So with the 4 images, where we can evaluate the infection very precisely. So at last, we also made some database. So if you scan this QR code, you will find the database that we made. It's a second database in the world. The first one is the MPS database in Pittsburgh, developed by the MPS consortium in the United States. And we also got the first society, the Organoid and Organs on Chip Society in China. It's under the Chinese Society of Biomedical Engineering. So if you scan the QR code, you can see that society. And we also established a company called AvaTarget. So for this one, you may have 2 ways to read it, like alpha target means it's a primary target, or avatar get means to get an avatar. So although there are very limited companies in China, but we're hoping that our company and others could really develop some organs-on-chip products in China that allows researchers and medical doctors to really use. So last, we thank the Ministry of Science and Technology and the Ministry of Education. And also we have to thank all the members in Professor Gu Zhongzhe's lab and all the friends that collaborated with us. Thank you.
Thank you, Mr. Chen. There is time for a short question.
Yes, please.
I think I don't see any question. Oh, sorry.
Very good talk, thank you. In the Chinese market, how soon when do you think we'll have personalized medicine? It sounded like you guys are already kind of looking at it in the clinic with organoid screening.
Thank you. In fact, it's quite interesting. In China, there are more and more doctors are interested in the organoid and organs-on-chip research. Now, even for the grant, they have a special grant code for organoid and organs-on-chip research. But the doctors are not only staying research only, but they are applying the IIT, the investigator-initiated clinical testings. So, as far as I know, in fact, there are more than 50 companies study the cancer organoids in China, and there are a lot of IIT have been started. More than 50 IIT have been started in China. So that's the reason I'm thinking like it probably need more regulation, but on the other hand, they only do the backward testing, not the forward testing, predicting what drug, but they are doing the backward testing. And the results are pretty good. In fact, it indicates like more than 80% of the drugs are very similar to what the in vitro tests are predicted, similar as Dr. Hans Cleves reported that. So I think it has a good chance in China to have more personalized medicine testing, and probably it will go faster than some other countries. Yeah. Thank you.